Hello my people. Today we are going to look at one of the most important processes in biology, mitosis. Mitosis is the way one cell divides its nucleus to create two identical daughter cells. You started as a single-celled zygote and became a complex person made of trillions of cells. That happened because of mitosis. We use it every day for growth, for repairing cuts, broken bones, and for replacing cells that just get too old to function. It is important to remember that while the cell cycle is the whole life of the cell, mitosis is the active step where the division happens. There are four main stages we will walk through today, followed by a process called cytokinesis. The four main stages of mitosis are prophase, metaphase, anaphase, telophase, all followed by cytokinesis. The first stage is prophase. This is the setup phase. During prophase, the nuclear membrane, the protective shell around the DNA, starts to fall apart, and the nucleolus disappears. Inside the nucleus, the stringy DNA called chromatin condenses into chromosomes. Chromosomes are the X-shaped structures on the diagram. These chromosomes were actually copied earlier during interphase, so they are ready to go. Finally, the spindle apparatus begins to form, which act like tiny cables that will eventually move the chromosomes around. Next up is metaphase. I like to remember this as M for middle. The chromosomes attach to those spindle fibers and line up perfectly in the middle of the cell. The fibers lock onto the centromeres, which is the center of that X shape. This stage is very short, but very important. If the cell messes up this lineup, the new cells might end up with too much or too little DNA, which can lead to serious problems. Metaphase is the cell's way of making sure everything is balanced before the big split. Once the lineup is perfect, we move into anaphase. Think of the A like it stands for apart. Those spindle fibers begin to shorten, which creates a pulling force. This force splits the centromeres and pulls the sister chromatids apart toward opposite poles of the cell. The chromosomes move to the far ends of the cell. By the end of anaphase, you have a complete identical set of DNA at each side of the cell. The final stage of mitosis is telophase. This is basically prophase in reverse. The chromosomes arrive at the poles and start to relax back into their stringy chromatin form. Two new nuclear membranes begin to form around the DNA, and the nucleoli reappear. At this point, the cell is done with the spindle fibers, so it recycles those structures to help rebuild its own internal skeleton. The internal skeleton is called cytoskeleton. Now there are two distinct nuclei in one single cell. Even though the DNA is divided, the cell itself still needs to split. This is called cytokinesis. In animal cells, the cell uses microfilaments to constrict or pinch the middle of the cell until it pops into two. However, plant cells have a rigid cell wall, so they can't pinch. Instead, they build a cell plate down the middle. New cell walls form on both sides of that plate, resulting in two genetically identical plant cells. This marks the official end of the division process. Where does mitosis fit into the cell cycle? Mitosis fits into the cell cycle as the final active division stage known as the M phase. It occurs immediately after the G2 phase of interphase. Interphase is where the cell has grown and replicated its DNA. Mitosis is the process of organizing and dividing the replicated chromosomes. It is followed by cytokinesis, which completes the cycle by splitting the cytoplasm into two identical daughter cells. Why mitosis is important? Mitosis is important because it is the fundamental process of cell division. Mitosis produces two genetically identical daughter cells from a single parent cell. It is crucial for growth, tissue repair, and asexual reproduction. It ensures genetic stability by accurately spreading chromosomes. Mitosis allows multicellular organisms to form, regenerate damaged cells, and replace worn-out cells. Without it, multicellular organisms couldn't develop from a single zygote. We wouldn't be able to heal a scratched knee or replace the millions of blood cells we lose every day. Mitosis is the reason our bodies can grow, regenerate, and stay healthy throughout our lives. Summary. To sum it up, the four main stages of mitosis are prophase, metaphase, anaphase, and telophase. During prophase, the nuclear envelope breaks down and the spindle apparatus begins to form. In metaphase, spindle fibers attach to the centromere of each sister chromatid when the chromosomes line up in the middle of the cell. During anaphase, sister chromatids are pulled to opposite poles, becoming individual chromosomes. In telophase, the chromosomes arrive at the opposite poles, decondense, and a new nuclear envelope forms around each set of chromosomes. Cytokinesis then divides the cytoplasm to complete cell division. The role of mitosis is to create two identical daughter cells because the new cells are necessary for growth, tissue repair, and asexual reproduction. This allows an organism to grow from a single-cell zygote into a multicellular organism by increasing the total cell number. This process also guarantees consistent genetic information across all new cells. For repair, it allows the body to regenerate damaged tissues and replace cells that are worn out. Thank you. Did you enjoy my presentation? Let's talk. Contact me at mckinleyallen@dcs.k12.al.us. Thank you.